and welcome to your next video on proteins and nucleic acids. So we have already talked about lipids and carbohydrates being macromolecules and how macromolecules are joined and separated. And now we're going to talk about proteins and nucleic acids. So proteins. Proteins are important in biological systems as control and structural elements. You may know proteins just because when you flex your muscle, you use the protein myosin in your muscles. Also, when you get sick, your immune system has these cool things called antibodies, and those antibodies are what keep us safe from bacteria and viruses. So proteins are very, very important in biological systems, especially us. They contain CHs and Os, which we found in our lipids and our carbohydrates, but now we're going to see nitrogens as well. And an example are enzymes. Some hormones transport bridges so they cross the cell membrane. Hair, hemoglobin, and blood, meat, and many, many more. And we're not going to go into every single one of those. So, proteins. The monomer protein is an amino acid, and there are 20 amino acids. However, humans only make 12. So how do you think we get the other 8? If you said in your head or out loud that we eat them, you would be correct. So if you look at this picture right here, this is a picture of an amino acid. It's the conventional depiction. And I want you to copy this down in your notes. It's very important. What we are seeing here is if we look over here, this is called a carboxyl group. You see the C, an O, and an OH. And that OH is very important. Remember our dehydration, synthesis, and our hydrolysis. That's going to be very important for that. And we have an amino group over here. And that's very important too, because we take this H over here and this OH whenever we do dehydration synthesis. So that is a very, very important part of our amino acid. Now, this is just a backbone of an amino acid. Every amino acid has this. The thing that changes on this amino acid, everything stays the same except this one group, and we call that the R group. On every different amino acid, we have different R groups. So this amino acid could have an H as an R group, or it could have a big long chain of carbons. Depending on the amino acid it is, it depends on what it changes. And we even see this new element in here, this nitrogen now. So we have special bonds in amino acid, in just proteins in general. Amino acids are linked together by joining the amino end of one group, so that H2N that we saw of one molecule to the carboxyl end of another using dehydration synthesis. Be thinking about what we talked about earlier with our dehydration synthesis and the removal of that water. So the formation of a type of covalent bond that is known as a peptide bond. And I know you're thinking about covalent bond and sharing those electrons. You're really thinking, okay, peptide bond. Well, peptide bond is very, very important because sometimes when we refer to proteins, we refer to them as a polypeptide. Remember, poly meaning many. Well, this is a peptide bond, so a very easy way to think of our proteins. Now, we have different ways of talking of our proteins, and we talk about our proteins in 4 structures. Our first structure is our primary structure, and our primary structure, we just talked about our amino acids, is solely putting those amino acids in place. Those amino acids being the different types of amino acids, and this is very important because if we miss one or we put the wrong one in, we could actually wind up with some kind of disease. People with sickle cell and anemia actually have the wrong type of one amino acid in an amino acid sequence. So this is very, very important. Now, the next thing that we do is we make a shape in our secondary structure. And this right here shows you the alpha helix. And we start to make a shape. And we're using hydrogen bonds. Do you remember where hydrogen bonds, we first learned about them? If you are thinking water, you would be correct. Well, we also use them in biological systems, and proteins are a very important biological molecule. Remember, we just said that. And so we use these hydrogen bonds to keep things in place. So we can have an alpha helix, but we can also have a pleated sheet, and that is called a beta pleated sheet. So that is our primary structure and our secondary structure. Be sure to draw really good notes on that. And then we come up with our tertiary structure. So now we're starting to fold into individual peptide bonds. So you see that we actually are folding in on ourselves. And if you actually have curly hair, you have a tertiary structure in your hair called a disulfide bond. Basically what's happening is your hair is bonding to itself, and when it folds in on itself, it's creating this special type of bond called a disulfide bond. In our tertiary structure, we're getting all kinds of special types of bonds. We're getting more hydrogen bonds. We're getting all kinds of things here. And so this is really giving our protein more shape. Well, at this point, we still have our polypeptide. We only have one protein. We only have one sequence of amino acids. Now, when we get to quaternary structure, we are adding more peptides or more polypeptides, meaning we have more sequences of amino acids. And what we are actually doing there is we are putting them together to make a usable protein. So you've heard of hemoglobin. Hemoglobin is what transfers oxygen in our blood. And that has four polypeptides. So its quaternary structure is going to have 4 polypeptides. So just because it is a peptide does not mean it is only one sequence of amino acids. It can be multiple sequences of amino acids. So a special type of protein that we talk about is enzymes. And enzymes are a biological catalyst. Catalysts are anything that will speed up a reaction by lowering the activation energy, and enzymes are protein molecules that act as catalysts in biochemical reactions. So what this does is it lowers its activation energy. So each enzyme has a specific structure. So does each protein, but it will only bond to a specific molecule, and we call that molecule the substrate. It then binds to another and repeats the process until there are no substrates left to break up. Substrate fits into the enzyme like a key fits into a lock. And what it's doing is it's lowering the activation energy. Anytime a chemical reaction takes place, you need a certain amount of energy to get the reaction started. What the enzyme is doing is lowering the amount of energy that is actually needed to get the reaction started. So this is actually helping us to make it go quicker. So if we look here, we see the substrate, we see the enzyme. The substrate fits completely into the enzyme. And so the enzyme can work on the substrate, and eventually we might have 2 small circles that we see there. So another thing I want to talk about before we go on to nucleic acids is, so we know our protein fits its structure. It won't work if it doesn't fit our structure. So what happens if it breaks? Well, it's not going to work anymore. If a locksmith comes and changes your lock at your house, your key is no longer going to work in that lock. So what's going to happen? Well, essentially, our protein is no longer going to work. And we call this denaturation. So I want you to write that down. Denaturation is when a protein is destroyed and no longer works. And this happens by exposure to excess heat, excess electricity, unbalanced pH, salinity, something of excess. And in actuality, you do this every day. You do this in your kitchen every day. So when you cook an egg, you cook some meat, you are denaturing those proteins so you can eat them. So just be thinking about that next time you are in the kitchen. Okay, so nucleic acids. Nucleic acids are polymers composed of monomer units. So we're still on these macromolecules of polymers. They're composed of monomer units known as nucleotides. And the functions of nucleotides are to store information and to play an integral part of protein synthesis. And later in the year, we will talk about exactly what protein synthesis is. Just know what it is exactly pretty much what it sounds like. We're making proteins. So a nucleotide right here consists of a sugar, a nitrogenous base, and a phosphate. So as you can see, we have our base, our sugar, and our nitrogenous base. Now, you know of two different nucleic acids. We have DNA and we have RNA. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Deoxyribonucleic acid has the sugar deoxyribose, and it is double-stranded, and it is in a double helix format. While ribonucleic acid, or RNA, is a single helix and single-stranded. Now, that being said, we have A, T, C, and G base pairs in DNA, and we have A, U, C, and G, and RNA. So make sure you write down those differences. We'll go over them in class. We're going to go over this entire video in class. So just to review what we just talked about, we talked about proteins, and proteins are a very, very important part of our biological system. We talked about our amino acids, how we get our amino acids, special type of protein bond. We talked about primary, secondary, tertiary, and quaternary structures. We talked about catalysts and enzymes. We talked about activation energy. We talked about substrates and enzyme substrate complex. And then we talked about nucleic acids. We also talked about denaturation. So make sure you have all that in your notes. Make sure you drew great diagrams, and we will talk about this later.